Hello everyone, I'm Brahim.
I'm Afnan.
Today we are going to present about the new technique which is MRM, multiple reaction monitoring. Our title is the use of multiplex MRM for the discovery biomarker to differentiate iron deficiency anemia from anemia of inflammation. This article was published in 27 June 2012 in University of Victoria. The outline for our presentation: introduction and aim, material, method, result and discussion, conclusion, peer review, and reference. Now we are start with introduction with Afnan.
Iron homeostasis in the body is very important for health. Too little iron leads to anemia, while too much iron toxic for the body. Anemia is condition that develops when blood lacks enough healthy red blood cells or hemoglobin. Hemoglobin is main part of red blood cells and bind oxygen. So, if you are abnormal red blood cells, low or abnormal hemoglobin, the cells do not get enough oxygen. Iron deficiency is treated with iron replacement, whereas iron replacement is not necessary for anemia inflammation. This, the importance of digesting need to compare both type in order to facilitate management or the underlying anemia.
The aim of this study is use MRM-based assay to identify panel of biomarker that distinguish between iron deficiency anemia and inflammation, and the combination of inflammation and anemia as a model of anemia of inflammation. The MRM coupled with SIS stable isotope label standard mass spectrometry is a powerful quantitation technique. The candidate biomarker protein of iron metabolism were used in this process. The next step is material and method. Actually, it is a long process, and start with the anemia and inflammation model. The total of 83 C5-7PL 6 to 8 week all female mice forming 4 experiment group. For this experiment, the inflammation model was generated by composed of inactive and dried Mycobacterium tuberculosis. The IDA mouse model by low iron diet combined with a weekly 0.5 ml phlebotomy for 3 weeks. Combined model of true IDA with inflammation model, low iron diet and with weekly 0.5 ml phlebotomy for the 3 weeks, and as well CFA treatment, which is composed of inactive and dried Mycobacterium tuberculosis. The normal model without treatment.
The next slide is show about sample preparation and bruising. Animals were anesthetized at the end of the experiment for blood collection. The mouse plasma samples were stored at 80 degrees until bruised. Diluted sample and ammonium bicarbonate buffer were denatured, reduced, alkalized, and then digested with trypsin for 16 hours at 37 degrees. 80 SIS peptides for 14 selected protein of iron metabolism were added to the digested sample. These samples were received in 2 batches, 23 in the first batch and 55 in the second batch.
The next step of material and method: LCMRM MS analyze. Sample were normalized to 1 microgram microliter by redehydration with 1.1% formic acid. A peptide mixture of 18 cis peptide corresponding to 14 protein were added and analyzed by nano ACIMRMS. MRM acquisition method were constructed using 3 ion pair per peptide with fragment ion specific, optimize the clustering, obtain and collation energy and detonation time constraint. Default collagen cell exits potential of 23 voltage was used for all MRM ion pair. The MRM data analyze used the to perform using 3 transition pair peptide and a multi-quant 1.1 with MQL algorithm for peak integration was used. This is the first result. Now we will talk about it.
3 MRM transition were monitored per peptide with one of 2 prototypic peptide being used for each protein. The MRM determined plasma concentration were then statistically analyzed to determine how effective each protein was at distinguish between normal animal and the disease models. Each protein was evaluated both individually, univariate and analyzed, and as part of multiple variant panel.
The next result is MRM analysis and statistic for biomarker panel of protein. The consideration of the potential for protein determined using MRM were statistically analyzed to study how effective each protein was in distinguishing normal and disease animal. The statistic method, the peak area rate of each protein were evaluated using two-way ANOVA for the effect of IDA inflammation and the combination between IDA and inflammation. P-value used to control the false discovery rate. Multivariate panel analysis built a panel of protein from the result of univariate test using type-wise discrimination analysis, in which have one out across validation was used to estimate the classification performance of each protein. The next step, evaluation of biomarker panel performance. The ability of different panel of protein classify sample within the 4 group was evaluated by LOCV. The principal component analysis, which is PCA, was used to illustrate the ability of multivariate panel to separate the 4 disease model group.
The example of the data made in this MRM-based analysis are illustrated for a protein known to be induced by inflammation cerebroplasmin. It is known that transferrin receptor 1 is well elevated in iron deficiency anemia, and the top graph A shows it is elevated in IDA mice.
Now we are moved to the summary of quantitation for all the 14 protein. The graph showed the quantitation result for this individual potential biomarker protein. It can be seen that the concentration level of IDA and inflammation plus IDA are overlapping among the different protein when using single biomarker are used, which is cerebroplasmin. Also, it has many of the biomarker show distinctly different level with inflammation alone compared with inflammation plus IDA. This suggests that as a combination of all this protein as a panel might be useful for distinguish the iron metabolic disorder. The next slide is univariate ANOVA to select the best candidate protein for biomarker panel to distinguish between normal and disease group. This slide as well, the p-value for pairwise comparison between 4 group using TUK HSD post HRC test. From this table and this figure, it can be determined that the plasma level of transferrin receptor 1 and APO4 were significantly different among a 4 group study when is the p-value less than 0.05, 0.001, as well the lactoferrin, hemopexin, and cerebroplasmin and heptoglobin. There is a significantly disease model effect when mean from 4 groups are compared by p-value less than 0.001. The next slide is batch effect using 3-way ANOVA.
This slide shows the batch effect analysis. Above A4 and lactoferrin showed a batch effect, whereas transfer didn't.
Now we are going to the last result, which is classification accuracy of individual and multiple protein biomarker panel and BCA. We can see in the left side the classification accuracy of individual protein biomarker. When the test sample are classified based on the single protein, the overall classification accuracy remain under 72% within the maximum accuracy attained for the transferrin receptor 1 and APO4. Among these three proteins, transferrin actually reached the highest performance within the group, 94% in normal group, and in general, there is no a single protein classifier that outperform all other within all group. On the right side, you can see the overall accuracy obtained by using a panel for 5 protein, which is transferrin, hemofixin, transferrin receptor 1, APO4, and MMP8, instead of the best single protein transferrin, improve the accuracy from 72 to 88. Moreover, the classification accuracy improved from 94% to 100% in normal group, from 50 to 72 in inflammation group, from 66 to 96 in IDA group, and for both inflammation and IDA group, the accuracy improvement from 79 to 83. In the slide in the B, the 3-dimension, you can see the ability of the selected 8 protein panel to discriminate the 4 analyzed group was evaluated further by the first 3 principal component from the principal component analysis, which is BCA. As the previously observed, the group with only inflammation, which is by blue color, is not well differentiated from the control group, which is black color. However, the other group, which is red and green, it has differentiate based on the multivariate panel. Now we are moved to the conclusion.
8 out of 14 protein, all this protein is expressed among 4 groups studied. All the 8 candidate protein were retained when analyzed as a multiple protein panel by BCA and demonstrated the differentiation of normal and disease state. Within this set of proteins, transferrin showed the best individual classification accuracy over the sample 72%, and within the NL group 94%. Compared the best single protein biomarker transferrin, the use of the compositive 8 protein biomarker panel improved the classification accuracy from 94% to 100%. In the NL group, from 50% to 72%. In the NL group, from 66% to 96% in the IDA group, and from 79% to 83% in the NPL plus IDA group.
Last step, peer review. The article is a part of special section entitled Understand Genome Regulation and Genetic Diversity by Mass Spectrometry. This has been cited 7 times in the literature. It has 1 tweet and 80 milliliter. The batch effect might have prevented to draw any conclusion due to the low abundant protein. Based on this finding, validation of the activity of this potential imported biomarker in human sample, it is in effort to differentiate between all the group, between IDA and inflammation. The explanation for the result is well elaborated to understand them clearly. However, some figure capture are missing in this paper. This is the reference. Feel free to ask question.
Thank you.
Thank you.
Thank you.